Hello, now that you understand a bit about what Mendel discovered, let's take this genetic stuff beyond Mendel. Let's look at some more complex inheritance patterns. For example, in this video, we're going to look at incomplete dominance. We're going to look at codominance, multiple alleles, polygenic inheritance, and effects of environment on gene expression. So let's get this started. So incomplete dominance, neither allele completely masks the expression of the other. So you've basically got a situation where you've got two alleles that are going, no, it's okay, you can express yourself. No, it's okay, you can express yourself. And they're giving way to each other. So what happens is in a heterozygous individual, they have a different phenotype than either parent. It's kind of like a blend. So if you have a pot or a can, and you've got this paint color, and it's being poured in, and it goes, well, I'll only fill it halfway, and then another pot or another paint color is poured in, and it fills it the other half, and you mix it up. You don't see either one of the paint colors that you poured into a can, but you see a blend of those two. This is incomplete dominance. Neither masks the expression of the other. You don't see either one in the offspring, but you see a blend of the two. So there is an intermediate phenotype, and this is sometimes called blended inheritance. So this is incomplete dominance. Please, please, please make sure you understand this, because this one and the next one are oftentimes confused. So examples of incomplete dominance are snapdragons. Snapdragons are actually a type of flower. So if we take a red snapdragon that is homozygous, so in other words, it would be a capital R, like we have here, because it's showing red and it's homozygous, and a white flower, which is also dominant. So what we would do is we would represent that homozygous individual as capital W, because it is not recessive. So we would not have a lowercase letter. So the red does not completely mask the white. So when we cross these two, when we cross a red with a white, we get this genotype, capital R, capital W. And this individual is actually pink. So we actually use we use different letters for the different characteristics because they are both dominant, but neither one is dominant over the other. Now it's rather interesting when you take a pink flower, or pink snapdragon flower in this case, and you cross it with another pink snapdragon. It's kind of interesting when you do this Punnett square. So we get a capital R, capital R, capital R, capital W, capital R, capital W, and a capital W, capital W. So remember, this is a pink crossed with a pink. So when we look at this, this individual is red. So two pinks can create a red. This one is pink, and this one is pink, and this one is white. So if you cross two pink snapdragons, hoping to get a bunch more pink snapdragons, that's not what you're going to get. You're going to get a 1 to 2 to 1 phenotypic ratio. Sorry about that. Phenotypic ratio. So you can see we have 1 red to 2 pinks to 1 white. So you can see that in this picture here. We have the parent generation where we started out with a pure red or homozygous red and a homozygous or pure white, and we cross those, and all of their offspring. The F1 generation is all pink. And you can see they sometimes represent this as C for color with a superscript of what that color is. So that can make this sometimes less confusing. So you can you can make all of these superscripted if you'd like. I will accept either way. I will accept you writing it this way or writing it this way. All right, so that was incomplete dominance. Good. Make sure you can describe that and give the example of the red snapdragon and the white snapdragon and the pink snapdragon, those flowers. So in humans, spacing between the eyes and wavy hair are example of incomplete dominance. So we can have anywhere from straight hair to extremely curly hair to just a bit of wavy hair. So this is an example of incomplete dominance. So next we have codominance. Please make sure you do not confuse this with the previous term. So codominance is this. It's similar. It's a similar concept to incomplete dominance because neither allele masks the other. Now the example that I give typically with codominance is if I were going to share my teaching responsibilities with someone, my teaching responsibilities to you with someone else. That other person and I are going to cooperate. On some days you'll see me. I will be fully expressed. And on other days you will see the other teacher, and that teacher will be fully expressed. Neither one of us masks the other, but both of us are fully expressed. So you can see this actually in several different examples, and I'll show you. So in codominance, both traits are shown in the phenotype of the offspring. So an example would be a barred rock chicken, and it results in the cross between a black and a white chicken. So again, our Punnett square is going to look like this. So if we have a black crossed with a white, we've got capital letters for both. So when we create a Punnett square, we have one allele type for each one. We have, excuse me, a Punnett square that looks just like it did with incomplete dominance, but our results we get are much different. We don't get a gray chicken, which is what we would get with incomplete dominance. With codominance, we get a speckled chicken. So you can see on this chicken, you can actually see the you can actually see the black parts of the chicken and the white parts of the chicken. This is codominance. In codominance, both phenotypes are expressed, and you will see both of them. You can also see the example in a roan cow or a roan horse. If I were to walk up to this cow and pull out a bunch of hairs, I could literally separate the hairs into red color and white color. So this is codominance. And this is where we get flowers that look like this. So we've got the red or pink shade that's fully expressed, and we have the white area that is also fully expressed. These are examples of codominance. The alleles cooperate. Some color one part of it, some color the other part. Both alleles are fully expressed, and you see them both in the offspring. Examples of codominance are also sickle cell anemia in humans. So that's a sickling of the shape of the red blood cells. You can see right here, sorry for that overlay of this, you can see right here we've got this shape of this red blood cell, whereas they're supposed to look like you see right down here at the bottom. So AB blood type is also an example of codominance because we've got an allele that says A and an allele that says B, and both of them are perfectly expressed or fully expressed, very much like singing an A note and a B note in a choir. You would hear both of those notes. Multiple alleles. Now, please don't confuse multiple alleles with the others. It's going to take some diligence to make sure you don't confuse these. So in some cases, an offspring inherits two alleles. In all cases, an offspring inherits two alleles, but in some cases, there's offered more than two alleles. So in some cases, an offspring inherits two alleles from parents where there's a choice of three or more. Now, each parent has two options, but they may not be the same two options. So let's take a look at this. Human examples are this. Again, going back to blood type, we have three different alleles for blood type in humans. We have I superscript A, I superscript B, and lowercase I. Now, each of the two parents have two of these three alleles, so they are going to have one of the two that they possess that they can give, but there are three alleles in the gene pool. Anytime you have more than two alleles in the gene pool, or in for that gene, this is called multiple alleles. So an example that we've got here are pigeon feather colors. So let me show you some of those. So we've got a red pigeon. Red is dominant to all the other alleles. So red would be generally expressed as a capital letter. Whereas we've got a blue pigeon is recessive to red, but dominant to chocolate. Now this is where it gets a little confusing because red is dominant to everything. Blue is dominant to chocolate, but recessive to red. Whereas chocolate is recessive to both. So if we were going to express these, or if we were going to write the phenotypes of these, red can be capital A, lowercase b, or capital A, lowercase c. So this can get a little confusing. So blue can be written lowercase b. The reason for that is because it's recessive to red. That's why it is a double lowercase letter, because it is recessive to something. But it's actually dominant to chocolate. So the chocolate would be the lowercase c. This is where this gets a little confusing, because in this case, B is actually dominant to C, but you don't see that very well when you write the genotypes. So we won't be right. I wouldn't be asking you for anything like this that's that complicated for multiple alleles on the test. So chocolate would be lowercase c, lowercase c. All right, so more examples of multiple alleles are the human blood typing. Human blood typing is fantastic for genetics. So there are multiple alleles. You can have the allele for A, you can have the allele for I, and A, the multiple allele. Let me stop. You can have the allele for I superscript A, I superscript B, or lowercase I. So we're not talking about the positive and negative factors at this point. That is called RH factors, and that's a whole nother gene. So we're talking about multiple alleles. There are three types of alleles for blood type in humans. So when we look at this, we have group or type A, group or type B, type AB, and type O. So antigens and antibodies are, excuse me, are what we're talking about with this. Talking about antibodies and antigens, you don't need to have a full understanding, or excuse me, you don't have to have full knowledge of this for the test, but this part you should know. So if you have blood type A, you can have this genotype I superscript A and lowercase I, and I superscript A, I superscript A. So both of these create an A blood type. So blood type B can be produced by I superscript B, lowercase I superscript B, I superscript B. Those are two genotypes that would both produce type B. Type AB can only be produced in one way. It's I superscript A, I superscript B. O can be produced in only one way, and that's a lowercase I and a lowercase I. Typically when I'm drawing my lowercase i's, I will draw them as a script so they don't look like something other than an I. So the blood types and the genotypes for blood you should definitely know. So let's do this. Let's cross a father who is heterozygous for B blood. So if he's type B, there's two ways that he can get that. He can be I superscript B, which we know he has to have. And his second letter could be lowercase I, or it could be I capital B. But here it says he's heterozygous, so that clarifies the issue. We know this individual has a second letter that is a lowercase I. We have a mother that is heterozygous for A, so she would have to have an I superscript A. That's what gives her the type A blood type. And her second letter would be a lowercase because she is heterozygous. So we're going to cross these individuals. So again, we need the gametes. So for this individual, we have I superscript B is one gamete, and lowercase I is the other gamete. And for this individual, we have I superscript A as one gamete, and we have lowercase I as the other gamete. Now, when we cross these, we get a 2 by 2 Punnett square. So we have 2 by 2. We have I superscript B, lowercase I, so we put each one of the information that's contained in each circle above each column. And then over here, we're going to put the other parent's information. And then we can fill in the Punnett square. And you can see this couple can have a type AB child, because that's this first one. They can have a type A child, because that's the genotype of this one. They can have a type B child, because that's this one. Or they can have a type O child, because that's this one. So you can see that this couple can have any one of the 4 blood types for their children. And you can do that by creating the Punnett square. So there's what we just did, and there's our Punnett square. So you can see this is type AB, so the phenotype is type AB. The phenotype here is type A. And if we say the word type, and type, that's actually the genotype. So type B. So those are the genotypes, and these are the phenotypes. So we have the genotype, the genotype and the phenotype. All right, the last one we're going to talk about is polygenic inheritance. Poly means many. So this is many genes that are contributing to one trait. So many genes. Don't confuse this with multiple alleles. Multiple alleles is talking about the alleles within one gene. This is talking about many genes contributing to one characteristic. So this is an inheritance pattern that is controlled by many genes. Okay, don't confuse this with multiple alleles. Multiple alleles is controlled, or is a trait that you can find more than 2 types of alleles. Okay, so if you're confused by this, come see me. So a trait controlled by a polygenic inheritance could have many phenotypes. Generally, we see these phenotypes resulting in a bell-shaped curve or a continuum, a range. So typically when we talk about these, we're talking about in humans, eye color, hair color, skin color, height. So if we took everybody in our class and arranged everybody according to height, we couldn't have the tall people over here, the short people over here. We'd have a continuum from the shortest person in the class to the tallest person in the class. And the same thing with the other examples as well. So examples of polygenic inheritance, again, are height and skin color, and eye color and hair color in humans. So if we arranged everybody by height, you can see we have an average that's generally somewhere in the middle. And you can see off to the side here, we've got the heights. So we've taken all these people and put them on bleachers according to their height. And then they would line up according to their height that's given over here. So we can do the same thing with skin color. And this is not a racist thing to talk about. This is just talking about the differences in skin color and acknowledging that we do have different skin color. So that is affected by many different genes. So this is polygenic. So gene expression, this is the last thing we'll talk about today. So gene expression relates to the presence or the dimension or the appearance of a trait for which genes are present. In other words, if you've got a gene that codes for something, it can either code for a whole lot of that thing or very little of that thing, and that's what we're talking about. So there are things that can affect that, or there are factors that can affect gene expression. There are things within a body or in the internal environment that can affect gene expression. Examples of that would be things like hormones or nutrition. So as you've been growing up, when you hit puberty, there are genes that were not expressed in your body before puberty, but when you hit puberty, hormones were activated, and now those internal factors in your body will then cause other things to happen in your body. We also have external factors or external environments that can change things like temperature, and I'll show you a couple of examples of those. They're actually pretty cool. These things, internal and external environmental factors, may have an effect on whether a gene is expressed at all, or if it is expressed, how much of it is expressed. And that's gene expression. It's the regulation of how much or how little, or if at all, a gene is expressed. So examples of gene expression are things like horns on a bighorn sheep. So some bighorn sheep have really big horns. They're bighorn sheep, their horns are not so big, and it's not just gender, but it's also environmental factors that come into play here. So the genes are the same in both of these sheep, but the phenotype are different. So what are the phenotypes affected by? Hopefully you said the environment in which this animal lives. So gene expression can also be affected by temperature, like for example, a weasel. So when you take a look at this weasel lives in very cold climates, whereas this weasel does not. And you can see the difference in the coat color. Both of these weasels have the same genetic information within each of them, but one expresses one coat color while another expresses another coat color. If you look up a Siamese cat, you'll also see the same thing in a Siamese cat. The tips of the ears, the tips of the nose, the paws, and the tail will all be a darker color. And this is because of those areas being a little cooler, being more vulnerable to cold temperatures. So that will actually affect the genetic expression of those genes in those cells, even within that one body, whereas the rest of the cells on the rest of the body will code for a lighter colored fur. It's actually pretty interesting. So that's gene expression. So again, please make sure that you understand these 5 different things that go beyond Mendel. There's there's 5 of them. Please make sure you don't mix them up. As a matter of fact, right now, at the very end of this video, turn your paper over, see if you can list the 5 and see if you can describe them without looking. So, and that's it. Have a great day.